Yeah, hello girls. I thought today I walk you through this PowerPoint about enzymes. I'm sure you've done some reading in the holidays, or at least I hope so, but it might still be helpful if I explain a few things to you. So first of all, what are enzymes? And we say enzymes are biological catalysts, and what that means, those of you who do chemistry might know what a catalyst is. A catalyst is a substance that speeds up a chemical reaction. And by saying that it's a biological catalyst, it means that it is a catalyst that is made by a living organism, and specifically they are proteins and they are synthesized by cells. So proteins in your cells that can speed up chemical reactions. Another keyword for enzymes is they have so-called active sites. So whenever in an exam you're going to be asked about to write about enzymes, you need to use the word active site. Active site is the area of the enzyme where it binds with the substrates, and the active site has a certain shape, and the shape is substrate specific, which means it can only bind with one specific substrate. So as a result, you actually have quite a lot of enzymes, I think about 2,000 different ones, that are responsible for catalyzing all the different chemical reactions that happen in your body. The chemical reactions happening in a living organism are called metabolism. So we also say enzymes control metabolic pathways. Metabolic pathways are reactions where one reaction, the product of one reaction is a substrate of the next, and you get a chain of reactions. For example, here in this slide we see a reaction that starts with substrate A, and that is then converted into B, which is converted into C, which is then converted into the final product D. And we've come across these chain reactions, for example, in respiration and photosynthesis, where reactions happen in a number of small steps. And since enzymes are substrate specific, for each of these steps you have to have your own specific enzyme. So in this example there are 3 steps which are controlled by 3 different enzymes, number 1, 2, and 3. Enzymes are named either by their substrate or by the type of reaction that they're catalyzing, and they always end on an ASE. So ASE, as soon as if you read anything with ASE, you know that this is an enzyme. So for example, a lipase is breaking down lipids, and lipids are fats and oils, and protease is breaking down proteins. Other enzymes are, for example, DNA polymerase, and that's an enzyme that is building DNA. Or let's say a hydrolase, that is a reaction that is hydrolyzing certain molecules, which means it is breaking it by adding water. Or a ligase, which is an enzyme that is joining molecules in the DNA synthesis. And enzymes speed up chemical reactions by lowering energy barriers. What does that mean? So, if you want a chemical reaction to begin, the reactants must absorb some energy. And that is actually quite fortunate, because what that means that the molecules usually don't break down spontaneously. So when you think of our body, all the molecules in our body are quite stable. You're not constantly falling apart. And to get these molecules in your body to react, you need to add a little bit of energy, and this is called the energy of activation. So in chemistry, what we often do if we want something to react is we heat the reactants. So we add energy by heating the molecules. But we can't really do this in a living organism for obvious reasons. So instead, what the living organisms do is they use enzymes, and these enzymes lower the energy barriers for the reaction to happen. So let's look at a graphic representation, what that actually means. So here we see a diagram, and we have this here represents the energy level, and you can see the reactants here. These are molecules, and they hold a certain amount of energy, and here you see the products, and they actually have less energy. So when something goes from a lot of energy to a little energy, that is usually something that happens quite spontaneously. However, for this reaction to start, these molecules need to get over an energy barrier. They need to be activated, and this means that this reaction does not occur spontaneously. And what the enzyme does, it is lowers this energy barrier, so it makes it easier for the reaction to happen. Here is another representation of the same thing. Here is the energy level, and here we see the progress of the reaction. So normally in a reaction we got the substrate here, or the reactants here, then they need to be activated, so the energy goes up, and then the reaction happens, and the final products hold less energy than the reactants. With an enzyme, this energy barrier is lowered, and the reaction can happen a lot more easily or a lot faster. So that's what enzymes do in our body. They speed up the reaction by lowering the activation energy or lowering the energy barrier. So how do they do this? How do they lower the activation energy? And there are basically two ways. Energy can either break down substances into their smaller components, or they can use smaller components and fuse them together. So if an enzyme is fusing something together, it provides a surface for the reaction. So what this means, let's say there are two types of molecules, A's and B's, and we want them to react and form a bond. And they're all moving around in the cell, and occasionally two of them meet and react. Well, what the enzyme does, it takes in one of A, it takes in one of B, it puts them in the right orientation, and thereby it makes it a lot easier for these two molecules to react. So it basically brings them together, and it brings them together in the right orientation. And that's what that means, provide a surface for reaction. If the enzyme is catalyzing a reaction where substances break apart, what it does, it takes in the substrate and then it puts it under tension, and that tension then weakens the bonds, and that makes it easier for the break to occur. Now, in your own time, later you can watch this animation. Because enzymes have a specific shape and their active site has a specific shape, they are selective, which means they can only react with a certain substrate. And that means that the type of enzymes present in your cell determine which kind of reactions are happening. Yeah, so a muscle cell will contain different enzymes than a liver cell or a brain cell, depending on the type of reaction that need to happen in each cell, it will hold a different set of enzymes. There are two models to explain how enzymes work. The older model is the lock and key model, so that orange blob here is supposed to be the enzyme, and this here is the substrate, and you can see this is a substrate made from two molecules, and the idea is to break these two molecules apart. So this substrate here fits perfectly into the so-called active site. So this here is the active site, and you can see how it fits in. And we say it fits like a key into a lock, and therefore it is called a lock and key model. And then the enzyme puts it under tension, and then the two molecules break apart. The more current and newer model is the induced fit model. And here you can see, so here is the enzyme, and you can see that the substrate doesn't really fit in perfectly yet. However, when it does bind to the enzyme, the active site changes its shape, it improves its shape, it molds around the substrate, and then it catalyzes the reaction. So we think by doing that, it does something to the substrate that speeds up the reaction. So for example, puts the substrate under tension, and then the substrate can break apart. So this is the more current model for how enzymes function. So here we see an enzyme. In this case it's called a sucrase. So ACE tells you that it's an enzyme, and sucrase tells you that it's an enzyme that is breaking apart sucrose, and sucrose is a disaccharide. So here is a sucrose molecule made from two molecules, one fructose and one glucose molecule. And we can see in number 2 how the sucrose molecule fits into the active site of the sucrase. So it binds into the enzyme with induced fit, and this is called the enzyme substrate complex. So it forms an enzyme substrate complex. Next, the substrate is converted into this product. For this bond to break, a water molecule has to be added, and this is called hydrolysis. And lastly, the two products, the glucose molecule and the fructose molecule, are released. And then the enzyme can start all over again, because the key thing here is that the enzyme does not change in the process. So the enzyme can be used over and over and over again. It's not involved in the actual reaction.